All right, so hello everyone. My name is Christian Plessl. I'm the director of the Paderborn Center for Parallel Computing, which is one of the hosts for this institution, for this event. This International Autumn School on CP2K GROMACS for Multiscale Atomistic Simulation event is a co-organized event. So from us in the HPC Center in Paderborn, but also from our contributing institution in the National High Performance Computing Alliance, NHR of Germany, and also with the involvement of CASUS, the Helmut Center for Advanced System Understanding. We're very happy to have you here at Paderborn University. The event was long planned, and we had a very strong response to the announcement. So the event is way overbooked, and we had to select the audience to not only to fit into the room, but also to make sure that there is the possibility to have real hands-on exercises. So it should be not like a pure method and teaching event, but also a practical event. And with this amount of participants, we are confident that we can still provide adequate support. Yeah, so we're very happy to have you here at Paderborn University. I already mentioned we have a HPC Center just across the street. I think there's also a tour offered where you can visit the facilities, and also Robert Schade will later on say a few words about our infrastructure, so I want to skip the details here. But suffice to say that we're one of these 9 national HPC centers at the Tier 2 level in Germany for providing HPC and AI infrastructure. And apart from providing infrastructure, we're also very strongly involved in training scientists, in particular young scientists, and use of state-of-the-art computing methods, and also collaboratively with our users further develop these methods. And this is what this event today is about. So we want to bring together people from different application fields working on joint methods or codes together with people working at the HPC centers and combining this knowledge to, first of all, teach you new methods, but also give you practical skills in applying them to your research problems. So with that, we have I'm very happy that we have been able to get very well-known, excellent speakers, leading method developers and researchers in the field to give these lessons. And as you see in the program, there's a combination of theory parts, lecture style, and then always hand-on exercises. As part of the event, there's also a social dinner that will be held tonight, so I think you should have received an invitation if you want to join that. And then I think that is, about the general logistics of the event. Maybe we can. Will you introduce the team later on, or so we'll see people with shirts from PC Square and name tags, and also Anna from Erlangen. Other people that will help you with the exercises. So if something goes wrong, please ask them for help. We're here to support, and then also give us feedback if you run a similar event in the future, how we can further improve it. Let us know what you liked, what you didn't like, and we hope that is a useful event for you. So without any further ado, I would like to keep this introduction short, hand over the word to Helmut Grubmüller to provide the first lecture in this hands-on autumn school, and wish you a pleasant week here in Paderborn. Thank you very much.
Yeah, good afternoon. It's a great pleasure to be here. First time I was here actually was as a pupil quite a while ago. Won a kind of a prize, and Paderborn University hosted a couple of colleagues and myself and treated us very, very well, I should say. So that's one reason I came back. And another reason, of course, that's a wonderful event here, which I'm happy and glad to support. I hope I can somehow match the background of the audience. So maybe you can help me a little bit with a show of hands. Who's in computing, high-performance computing mostly? Okay. Let's go through the ranks. So physicists, okay, few, yeah. me too. Chemists, maybe, oh, quite a lot. Yeah, great. That's why QMMM will be coming up. Biologists, maybe. Okay. Mathematicians. Hoi, that's a wild mix. Wonderful. Did I forget anyone? Philosophy? I don't know. Okay, so got everything. So that's really a great mix. I'm happy with such a mix, and I try to have a few things for everyone, more or less. Yeah. So if there's a little bit too much biology for the physicists, sorry for that. If there's a bit too much mathematics or physics for the biologists, I think we'll kind of balance. If I'm too fast, if you don't understand something, please, we are not so many here, so please raise your hands or speak up and slow me down or ask a question. Even if you interrupt me, no problem. I got a couple of minutes more, so we can really enjoy a very informal setting here, I guess. All right, so first of all, my thanks, of course, also go to the organizers for setting up a wonderful workshop here, which I think you will be profiting quite a bit. And also my thanks go to my department who did all the work I'll be reporting on. So, and particular thanks maybe also to those few gentlemen, 4 of them who will be doing the hands-on, the exercises this afternoon. So they are not yet here, but we expect them to arrive shortly. Right, so I guess what I'm trying to do is, it's an introduction into force field molecular dynamic simulation, so there will be no chemistry, right? So by which I mean no breaking or formation of bonds. Maybe a few at the very end when we talk about constant pH very, very shortly. But otherwise, I see my role here is first to show you, of course, the basic concepts, but of force field-based molecular MD. But then also to motivate you to why should we do all that and what is the use of that. And so we will have a little bit of biologically, let's call it applications. What can we do with that? Examples of questions in biology, structural biology mostly, which we can address with these methods. Since there are mathematicians, computer science, and physicists also there, the biologists and chemists may lean back a little bit for the first few slides, you know all that, but let's try to get you a bit into proteins and macromolecules, biologically macromolecules. And then I'd like to also show you a bit of methods development, not so much in how to make GROMACS, which we will be using as fast as possible. We will do that anyway, but for example, to look at a bit entropy and free energy calculation. So that would be the plan, right? So, and I have a long list of slides and topics, so we just go as long as you guys stop me. When should I stop, by the way? At?
According to the agenda, 3.
3, okay.
So you can take your time.
Yeah, wonderful. So, looks like, yeah, still 4 minutes. Donation. All right. And so let's start from the very basics. Our objects of interest are proteins. And just right now, for lunch, I guess you have digested a couple of them, which is kind of a pity, right? So because it's like these wonderful cars, like Ferraris or what know I, Porsches, and you just recycle them, right? So you put them into the recycling press, tear apart everything. You can do that, and this is what you're gonna do with proteins if you eat them, and you digest them into their smaller pieces, amino acids. But somehow it's a pity, right? It's much better to let the Porsche drive and do its stuff, and the same with proteins. So besides just being nutritions, they are macro machines, nano machines, I should say, micro machines. They perform tasks in your body, and essentially every task in your body, almost every task, with very few exceptions in your body, is actually carried out by proteins. And so I was once told for every talk or so, people take home maybe one information item, one sentence. And so let me choose that sentence for you to take home. That would be this: protein are machines, right? And they perform tasks. And maybe if you allow me a second sentence, the way they perform their tasks again, in most of the cases, is by motion, like our macroscopic machines, right? They move in order. If you think about our Porsche, the engine of this Porsche again, it's moving pistons in the cylinders and moving crankshafts and valves and you know what. And these motions have to be highly concerted, highly controlled in order for this machine to perform the task. And it's exactly the same for proteins, right? Let me give you an example right now, just that we know what we've been talking about. So here, that is one of my favorite machines. It's called F-ATPase or F-ATPase. It's in your mitochondria, and if you have paid attention in biology classes, the mitochondria are the power plants of your cells. They produce the ATP for you. Now, the ATP is the fuel of your, about every or most processes which need energy in your body, like muscle contraction or synthesis also of certain chemical compounds. They require a fuel, and this is ATP, adenosine triphosphate. And these machines here synthesize this fuel for you. So actually they are more the fuel plants rather than the power plants. And they do that by exploiting a proton gradient between inner and outer membrane. So this is the inner membrane of the mitochondria, and these proteins, they sit right in the membrane. And from photosynthesis in plants or from your respiratory cycle, the primary energy buildup is a proton gradient, a concentration gradient or pH gradient, if you want, between this and this side of the membrane. And this machine, this part of the machine, uses this gradient very much like an electro engine, an electromotor. Protons can't go through the membrane, but they can go through this channel here. And every single proton which goes through this channel turns around this subunit here in the membrane by just one subunit here, right? And so if you have a proton flux through, this thing will rotate. And when I first saw that, I was totally surprised because my biology teacher at school told me nature, evolution, everything except gears and wheels, right? And apparently he was wrong, right? So that's wonderful, gears and wheels. He also mentioned the laser. He may have been right with that one, but gears and wheels we do have in our bodies, just not on a macroscopic level for certain restrictions. All right, so this rotation also rotates this orange part here. That's called the gamma subunit. And this orange part, you can see it here from the top, rotates inside this bearing, if you want. And because it's not perfectly round, it's kind of eccentric, the rotation induces conformational motions in these subunits specifically, right? And these conformational motions changes, propagate like a domino chain towards these positions here. These are the active sites. This one is empty. You can see the trifold symmetry here, right? And this is loaded with the product, ATP, which is about to leave the binding site. And this is the educt, ADP, and the phosphate comes in, and then this is synthesized. It's really amazing how this machine does it, considering how industry does it. Yeah, you can also buy ATP, of course. It's quite expensive. And in industry, what they do is they also mix together ADP and phosphates. But then, if you recall, the phosphates and also the ATP carry a negative charge, and negative charges repel each other. So you have to need energy force to bring them together. And what they do in industry is they heat it up, put it the kind of temperature motion of the molecules, makes them fast enough to overcome this repulsion. But of course, it has to occur just at the right orientation of the molecules. If not, go away again. And with the right speed, not too fast, not too slow. So you can see, and then they bump into each other and form this chemical bond, which forms the ATP. And you can see this is not elegant, not at all. Yeah, it's just randomness, and occasionally it works. Most of the time it doesn't. That's how industry does it. How does nature do it? Well, it just binds ADP and ATP in an energy well. So the binding pocket is kind of an energy well. So both of them, imagine two charged balls running down a well. Because of gravity, they overcome their repulsion. Here it's the chemical affinity, which overcomes the repulsion. So here they form the ATP kind automatically. But of course, also nature isn't able to overcome the first law of thermodynamics, energy conservation. So somehow you need to spend energy. And here this machine spends the energy in getting the product out of the binding pocket. Remember, we are at the bottom of an energy well. We have to get it out again. And this is precisely where this mechanical energy of this rotating gamma stalk actually changes the conformation of this binding pocket, just such that the affinity goes from high, means low free energy, to low affinity, means high energy, and the whole thing is getting out of the binding pocket. That's where the energy for synthesis of ATP, 13 kcal per mole, is actually spent. So I'm just explaining a little bit more detail here. First of all, to make you a little bit excited and curious about these nanomachines, right? They can do that much better than we can do it. And you will agree, this is certainly much more elegant, and it's really a nanomechanics which does the whole thing, which compared to what we do in our industry. So there's a lot to be learned from that. But irrespective of that, I think you also can see that there is a close analogy, a similarity with the function of an Otto engine again. So it was not an incident that I mentioned a car initially. So such a 3-cylinder Otto engine works about as this machine also works, right? We have the crankshaft, the rotating subunit here. We have these connections here, Pleuelstangen, no idea what this is in English, which move here the pistons in the cylinders. This is the conformational motion which I was mentioning in our analogy. And then there is the ignition, and that is the fuel, in this case ATP, being cleaved. Now I changed suddenly the rotations, reversed the reaction direction. I was always mentioning synthesis. Suddenly I'm mentioning the reverse, the cleavage of ATP, hydrolysis, and that also, in fact, can now make this gamma rotation go, right? So you can feed this machine with ATP, and then it cleaves it, and this energy is used to spin this gamma unit very much as in your Otto engine. I tried with my car the synthesis direction to push it, right, and waiting for fuel to come out. It didn't work. The reason being, my car is irreversible. It has a very low efficiency, like 20-25% for an Otto engine. And that's why this pushing and getting fuel out of it doesn't work. Here, the efficiency is near 100%. Again, much better than we can do. And therefore, it's fully reversible, and you can drive it in both reactions. Okay, so much about these machines. Our aim is really to understand these machines from first principles. And you also see here, maybe a good idea also to mention that here, there is a little bit of chemistry, namely the synthesis of ATP in the binding pocket, but all the rest doesn't involve any chemistry. And that is why force field simulations can help us a lot to understand what's going on there. The only chemistry going on here is this formation of a single bond, and we can just fake that in molecular dynamics force field simulations, right? We just form the bond and pretend now and see what's going on. But the most important reactions, if you want, no chemical reactions, but conformational changes, are the rotation of the stalk and how this energy is transferred to the binding sites and how the affinity changes. This is a non-covalent binding, I should say. And so all of that, all the interesting stuff here, except the single bond formation, can be done with force field, right? And why should we spend so much computer time and computer cost in QMMM if we only need it, if at all, at a very small region? And of course, you will learn about QMMM also later on in this course. Okay, just to give you another very short view of another machine, which we are working heavily on in my group, that's a ribosome. And the ribosome is kind of key, of course, as a very central machine in your body, because it synthesizes all the proteins, all the other machines, from the blueprint of the RNA, which again is a copy of the DNA. And so all the proteins are made by these machines, and also those, as you can see, undergo a lot of motion. That's not a simulation, that's from cryo-EM, cryo-electron microscopy. But you can again see how extremely dynamic and this machine is, and what an important role conformational motions actually play. And this is mostly true for most of the proteins which we study. Here is again a whole zoo of proteins which you find in your mitochondria, or also in plants. Again, our synthase again, but the whole thing is a whole chain, a factory of machines where one machine outputs into another machine which uses that and an input and forwards it to the next, and so on and so on. So just to give you a kind of a broader view, and there's really many of those machines. Currently we know from experiment, I think like 200,000 different protein structures, so really many of them. And of course, many of you will know about AlphaFold, so now we have an explosion of protein structures, which in most cases will be very close to reality, although we never know that. Okay, one final kind of general word in my introduction. As most of you will know, proteins are polymers, they are chains, and these chains are built up of 20, sometimes 21 different amino acids. Those are those 20 amino acids. And it is the sequence of these amino acids along the chain which is encoded in the DNA and also which determines how the protein, self-organized, folds up into a three-dimensional structure, like the ATPase I have shown you, and thereby performs its function. So the structure is necessary for the protein to perform its function. And this is encoded in the sequence. And this is exactly what AlphaFold does for you now, right? So to get from the sequence to the structure, not necessarily to the function. And again, just to rephrase, the main aim of our work in my group and in many other groups, and maybe of you, is really to understand these functions in microscopic terms from first principles, right? For the biologists, that is a little different from the models you are being used to, right? You are used to models which somehow come ad hoc and then have a few parameters which you want to fit against to experiment, right? That is what I would call a top-bottom or top-down model, right? Whereas here we are talking about bottom-up models where we start from fundamental physics principles and work our way upwards to biology. Okay, so that was the general intro. Are there any questions on proteins, on whatever? No? If you don't ask questions, I assume everyone understands. Yeah, please.
How did you get the rotor spinning in the simulation, in the ATP Synthase simulations?
We only looked so far, or mostly looked so far, in synthesis direction to synthesize ATP. And the spinning, this is a special potential actually built into GROMACS. We built it in. Carsten Kutzner, who will be here this afternoon in the practice, actually did that. And very briefly, you grab the whole gamma subunit with an artificial potential which rotates. Okay. Very gently, not to break it, very slowly, and that is how we rotate it. We don't simulate the proton flux in the lower part, and so we just gently grab it and turn it around. Okay. Okay. So my next chapter would be on what do we do actually when we do molecular dynamics simulations, again, force field-based simulations. And let me use the quite not so recent example, but I think it illustrates still very nicely what we do. That's Aquaporine. It's a passive water pore. It's just a hole where water molecules, one after the other, can pass and nothing else. You have them in the kidneys, in the islands, in the brain, in red blood cells, etc. So everywhere where you need to get water and only water out or inside certain cells. And I'm showing that because it nicely illustrates what we are doing here. So I will my explanation that will be in two steps. One is a hand-waving Mickey Mouse picture, and the other will be a bit more detail, right, for the physicists. So the hand-waving picture is what we do in such a simulation. We take all the atom positions we need to get from experiment, right, for crystallography, for cryo-EM these days, or NMR, of our molecule of interest, like the aquaporine, which you can see here shining through in colors. We put that into the computer, into a large array, XYZ, just normal coordinates. And then we ask the computer to calculate all the forces onto each single of those atoms, which exert all other atoms on this particular atom. And this we do for every single atom, right? So indicated here. So if we want to know, for example, the force on this particular red atom, of course, most of the atoms carry at least a partial charge. So we need to calculate the electrostatic forces from all other atoms onto this one atom. You can already see that is a double loop, right? That will be quite a bit of work. But then there is also van der Waals forces, which is a weak attraction. There is repulsion Pauli forces, which prevent all of us from just falling through the floor. Good that these forces exist. So they prevent atoms from moving through each other. There's also implemented. And chemical binding forces, yeah, chemical bonds. I know bonds are not springs, but it's a good approximation, at least at room temperature, and at least as long as bonds don't need to break. And so on and so forth. So we put all these forces into our GROMACS program, or whatever program you want to use. And then from these forces, we can go back and ask our good friend Sir Isaac Newton what would be the acceleration. And he will be telling us, and then we get an acceleration, and that changes the velocity, and the velocity changes the positions of all these atoms, right? After they have changed a little bit, of course, we have to say stop, because all the forces will have changed. Right? Imagine such a chemical bond, which is probably a bit extended. So the force will quickly change, very quickly, in fact, after a couple of femtoseconds, 10 to the minus 15 seconds. So we have to go back and ask our computer, oh, please, can you calculate all the forces again for us? Yeah, we'll do that. I'm a patient computer. And so that gives us another round of accelerations, velocity changes, and you can see where that goes. We repeat this loop over and over and over and over and over and over and over again until we accumulated a few weeks of computer time, right? And that will give us, for large systems, a couple of microseconds of simulations, right? So 10 to the minus 15, 10 to the minus 6 microseconds. That already is something like 1 billion of integration steps. So you can see that's quite a bit of computational work. But the principle is rather simple. And going back to how we calculate these forces, that's a little bit more integrated. But the principle is really simple, a double loop over all the forces. And for each integration step, then another loop outside that one. And just to also drive your intuition, this is exactly what also NASA does, or ESA, when we want to send space probes to Jupiter or Saturn or something, right? To calculate the trajectory around all the moons of Saturn, for example, in this case, you also go stepwise forwards, right? And you do the same thing. And coming back to our Aquaporine, what you get out of it is a raw data, if you want, is a molecular movie. Super slow motion, obviously, right? So we have, like, in this case, 20 picoseconds per second movie. And you can see the whirling of the water molecules. That's how it looks in your coffee cup in ultra-high magnification. And you can also nicely see the water molecules going through the channel. Actually, we would have to wait a few minutes here to really see them to go through the channel. But rest assured, we counted them, and every nanosecond or so we saw a water molecule coming through the channel, which is nice, because this is exactly what's been measured in terms of permeability to water of the Aquaporine. So that's a first check against experiment. And we do that over and over again, check against experiments, to really make sure that we are calculating accurately and close, and therefore are close enough to reality. This is, by the way, lipid membranes, lipid heads, charged and uncharged, hydrophobic tails in green here, and yeah, and the protein is here in blue. Whenever I'm showing a movie, by the way, most of the cases, here not, but most of the cases I'm leaving off the water. That just means I'm not showing it. It's fully in the simulation, and I think actually we may discuss that, but I think including the water is very important, right? There is so-called implicit solvent models, and they work solar, but I think I feel much more comfortable if I have the water explicitly, by which I mean molecule by molecule in the simulations. So I mentioned, yes, please.
Does it matter which water model do you use, like 3.4, 3.5?
Yes. It does vary. Luckily here, not so much. What we routinely do, I don't recall what model we used here. I think it was TIP3P, a very simple one. But what we do, whenever we do a simulation, we do it with different force fields, both force fields and water models, and hope that the results don't change so much. If they do, we have to rethink the whole thing. In this case, I think the dependency was not so large. We also predicted a number of mutations on basis of this, and predicted how much it should change the permeability, and also that worked out. It was measured subsequently. So that's something I can really recommend. The danger is, of course, it doesn't work out, and then you have to throw away everything. But yeah. But talking about accuracy, why am I actually insisting, or why am I such a fan of comparing to experiment all the time? Before I said it's from fundamental laws, right? And if everything comes from fundamental laws, it should be accurate, right? When we talk about these space probes, they don't compare to experiments so much, right? Because they know gravity. Okay, for the experts, solar wind is an issue. But nevertheless, they are pretty accurate. They are so accurate that people were carried to the moon and maybe at some point to Mars, I don't know. And they put their lives on the accuracy of that one, right? With molecular dynamic simulations. But why is that? And the reason is that for our space probes, our force field, in this case gravity, is known very precisely, right? Whereas our force fields are not so accurate. You indicated with your question already to this very fact. And the reason for that is that this system is much more complicated, and actually we are very much forced to do a series of approximations. NASA doesn't have to do that, almost, but we need to do that. And now a bit more for the physicists, I would like to sketch very, very, very briefly and very superficially the 3 levels of approximations which we actually need to do. Just that you know that we are not necessarily arbitrarily accurate. Yeah, we always have to check. And if you think back in your, I don't know, 4th semester or so in physics, and I would have asked you, calculate the motion of all the atoms in a protein, and I would have mentioned Newton, you would have looked at me with embarrassment and say, wait a minute, that's all quantum mechanics, right? So how can you dare to use Newton's mechanics equations in order to calculate the motion of atoms? Just use the Schrödinger equation. And this is actually what we should do. So if we really want to calculate accurately the dynamics of a protein, we would choose this equation, right? So that's the Schrödinger equation. And this is what you will do later on in this course, but not for force fields, not directly for force field simulations. Actually, you will not solve this one. We will solve the time-independent one, but we'll get into that. So that's the time-dependent one. And that contains, of course, the psi function. And the psi function contains as dynamic variables, in this case, all the electrons of the system, that's lowercase r, and all the nuclei of the system. And now I'm doing an approximation, so I will just treat the nucleus as a point charge, but that's good enough. So these are the positions of all the nuclei. So this is like 10, 12 times more degrees of freedom particles than we want to have. We want to have just the atoms and not the electrons. And this is the Hamiltonian for the experts. And one thing which is very often, when I ask students in examinations what goes into this Hamiltonian, at this level, they tell me, oh, it's this force field, right? Oops. Oh, something happened. That looks like as if my internet connection. It's either Rome. I think the internet still works. Let's see. Yeah, it's not the internet, so it's about Zoom. And now it's maybe back.
Could you please
Try to reconnect or share?
Yes.
Let's try sharing. And I need to use this one. And start. Oh, wait a minute. There. Yeah, perfect. Okay, so.
Could you please the small, click the small, this one.
Like this?
Yes, thanks.
Some like that. Okay, great. So I'm asking my students, what is this Hamiltonian? And they tell me, oh, it's the force field, electrostatics, bonds, angles, dihedrals, Pauli, van der Waals. And that, if they tell me that, that reveals a deep misunderstanding of what's going on here, right? Because all there is here, as interaction, we are on a deeper level now, right? Is the interactions between electrons, electron nuclei, and nuclei. And the only relevant interaction here is electrostatics at this level. There is no force field, no van der Waals, no nothing. All that is a result of the quantum mechanics coming out of this equation, but going into this equation is just electrostatics, Coulomb interaction. So this Hamiltonian, and of course kinetic terms, and so this Hamiltonian is super simple. It's just Coulomb, and you can easily write it down. So this equation is actually super simple, but super hard to solve for more than 2 or 3 atoms. And we talk about 100,000 up to 10 million atoms, so quite a bit. It's actually so computationally hard that, again, that not only 1 but 3 levels of approximations are required if we want to simulate large systems for microseconds. If you want to simulate a small system for a femtosecond, go ahead with this equation, no problem. But other than that, we need to approximate. So what do we do? First, physicists will know the Born-Oppenheimer approximation that just observes that electrons, being lighter than nuclei, much lighter, move much faster on average. Yeah. For the purists, quantum mechanics expectation values are faster. And therefore, what we can do is we can pretend that the nuclei, moving very slow, just are arrested in time for a little bit, right? So let's just say they don't move at all, and then let's worry about the electrons, how they equilibrate in the electrostatic field of the nuclei into their ground state. We always mostly talk about ground state, except light is involved, right? So at room temperature, most things are in ground state as far as the electrons are concerned. And so all we need to do is, for a given position of the nuclei, we need to calculate the ground state wave functions for the electrons. And this is done with this nice equation. And notice that there's a semicolon here, which means these now are parameters. As I said, we put the atoms we put on hold, and they are not dynamic variables anymore. They are the parameters. That's the meaning of this semicolon. And the only dynamic variables are the R's. And so what we get as the ground state energy here is actually a function of the parametrically of the atomic positions, right? So that's this E. And so from that, we can move the nuclei a little bit. And again, we should do that quantum mechanically strictly. But here, this motion we describe classically. That's our second approximation, right? And how can I dare doing that? Well, I would never do that with liquid helium at 1 Kelvin, because then some of you might know suprafluidity is a quantum phenomenon, and we won't catch them here when we do the classical approximation. But at room temperature for biological samples, which are embedded in a condensed phase, which destroys the phases all the time, actually it turns out that the classical approximation is pretty good. Surprisingly good, I should say. And I could take another hour why that is, and what we don't know about why that is. But again, comparing over and over again to experiment tell us that this approximation is mostly good enough. Again, for the experts, if you want to calculate the heat capacity, that might be a problem because of the bond vibrations being here also in the quantum mechanical ground state. But other than that, it's a very good approximation. Good enough for us. So that's number 2. And then, yeah, what we now, in this scenario, with these 2 approximations, what we would need to do is to calculate the ground state energy here. Also the derivative, because we want to put that into our Newton equation that gives us the forces. That's also a little bit non-trivial, but you have to look up quantum mechanics for this, Ehrenfest theorem. But once you have that, classically the positions of the nuclei for a femtosecond or 2 or 3. And then again, you would need to solve this equation here for a new ground state energy. So the electron wave functions follow smoothly and almost instantaneously the motion of the nuclei because of our validity of the Born-Oppenheimer approximation. Okay, so that would be still quite a bit of an effort, but this is exactly what you do if you do now quantum chemistry, right? If you, tomorrow or so, or at least with part of the system, if you do QM-MM, right? The QM region of the system, you will do exactly that. Every integration step, you do some density functional theory or whatever ab initio and calculate this value and the forces. And you will see, I hope I don't step on anybody's feet, but it will be significantly slower than our force field simulation. I guess this is still true. Maybe 2 orders of magnitude, maybe 3. It depends on the method you're using and the accuracy requirements. You can also be 10 to the 10 times slower if you want to be super accurate, but not much faster than 2 orders of magnitude slower. And so there comes the wish to still accelerate the simulations. And the idea is, again, pretty simple. We have here a high-dimensional function, and we need to approximate that. But first of all, we need to tabulate it, right? so we don't have to calculate it over and over again in each integration step. But once we tabulated it, we want to find a fit function which kind of fits these energy, ground state energy function. And this fit function is exactly the force field, right? That is also something which is, this connection is often not made by students, right? So they somehow have in their heads the ground state energy, that's a quantum mechanical thing, and then they have this force field, which is a kind, with all these terms, bonded, dihedrals and so, and this is the so-called classical thing. I have no idea why this is called classical. It's a fit function to a quantum mechanical function, ground state energy. So that's another semantic mix-up. The only classical which comes in is the equation for the nuclei. The electrons, by the way, also for your background, they are not treated in classical approximation, not at all, because we solve the Schrödinger equation either beforehand or during the simulations, or we use a fit function to the Schrödinger equation. It's no classical approximation. Okay, great. Having said that, I'm just emphasizing that so much because over 20 years of examinations, I'm not able as a teacher to really convey this message properly. Half of the students still miss that, so I'm trying hard to overcome that. Okay, and yeah, that's this empirical force field. I should also mention something here on the side. I was saying what we need to do here is to approximate a high-dimensional function, this ground state energy. And again, let me repeat, high-dimensional, if we talk about 1 million atoms, these are 3 million XYZ coordinates. So it's really super high-dimensional. And if you want to approximate a super high-dimensional function these days, what you do, you use machine learning, right? And this is exactly coming up in our field here. Rather than using this empirical force field, well, why didn't it show up? Whoa. Oh, am I frozen? Again, what's going on here? No, now it works. Okay, good. Right. So this thing, rather than using an empirical force field, where we use just simple functions from which we expect or think for some physics reason that they might provide a good fit function, rather than that, we might learn the force field, right? Carry out a whole bunch of quantum mechanics simulations to cover this high-dimensional configuration space as in the relevant regions as evenly as possible, and then use deep learning to approximate this instead of this analytical function. I think that's a good idea. It's not just yet fully finished, but many groups are actively working on that. And having said that, I should also probably say that on this level, we also have the option to do some deep learning solving the Schrödinger equation. I'm not sure if that will be coming up in the next days, but just to flag you, this is also a great opportunity to use deep learning. And finally, of course, maybe that's the least promising currently. You might think about, and people have thought about, rather than propagating in very small integration steps, like a few femtoseconds, maybe we can learn this propagator, again, from feeding in many molecular dynamics simulations into a neural net, and then maybe learn a 1 picosecond step rather than a few femtoseconds. That has not yet worked very nicely, I should say, although down here, that seems to work very, very well in many cases, but this is maybe something which we need to work further on it. It seems to be the hardest one, not so good for machine learning. All right, but just to prime you, so there's ample chances to also apply machine learning, and we live in exciting times because that is now really taking off here. Okay, having said all that, I think it should be also pretty clear that beyond these fundamental approximations which we have, our method, force field-based simulations, also has some kind of practical limitations, which just come from available computer technology and how fast our current, or how slow still our current computers are. And that limits timescales to something like microseconds. So just to give you an idea, if you want to simulate a reasonably typically sized system of a few 100,000 atoms on a GPU, by the way, I saw there's a GPU session later on. So GROMACS runs wonderfully on GPUs. All I'm showing is run on GPUs. Yeah, so if you want to use that, that gives you per GPU about, let's say, 100, maybe a few hundred nanoseconds per day, right? So if you want 10 microseconds, it's a couple of weeks or so, a week. Or you hook up a few more graphics cards. If you want to go to milliseconds, that's about the duration of your PhD thesis. So maybe you don't want that. So you can see where this limit comes from. But we are also living in quite exciting times, I should say, because that's where we are just now entering the biochemical timescales, right? So I was mentioning this water passage, that was an exception, so nanosecond. That's one of the very few biologically relevant processes which actually go that fast. But most of this stuff occurs in these timescales. Example, the rotation of our gamma subunit, if we drive it as fast as we can, is about 100,000 revolutions per second. So there we are, 10 microseconds. Yeah, 10 microseconds per revolution. We are right in there, right? We can do that these days. I showed the ribosome. The ribosome is able to attach about 20 amino acids per second to the growing nascent chain of the polypeptide. That's a bit out of reach still, right? So that's a few milliseconds, which we would like to do. But just to give an impression of how fast or slow things happen. And I'm not going into protein folding, but that can be even seconds or minutes, right? The fastest folders are microseconds. We can do that, right? So we can really fold ab initio if you want. No, I shouldn't say ab initio. That's for the quantum mechanics people. First principles, I should say. Otherwise, they get angry. So from first principles, yeah, you know them. You are not ab initio. Yeah, so we are first principles. And you can do protein folding from first principles, provided you have a powerful computer which can run long enough to fold the protein. There's no fundamental obstacle here. And the force fields seem to be good enough for that. Okay, but it's pretty clear this thing doesn't suffice. You need rather these things. Okay, I was already emphasizing the need to always compare to experiment. I would like, before I show you the first application, I would like to discuss a little bit one of my favorite comparison to experiment, and this is to atomic force microscopy experiments, and they become fashionable in the 90s, last century. What people do is, in this case, Helmut Grubmüller, this is the cantilever or few micrometer-sized spring, metal spring, if you want, which is the core of an atomic force microscope. And the tip of this cantilever, which is super sharp, is connected via a few polymers to the ligands of these proteins here. These ligands are biotins. That's just a vitamin, small molecule, 20 atoms. And this protein here to the binding pockets, here is a zoom to this protein. It binds highly specific and with high affinity this ligand into the binding pocket. That's all it does. This protein is called Streptavidin. And so a typical setup you would have is you connect also a few biotins here to a surface, like a small bead or something. These are mostly sugar polymers here. And then if you have this cantilever loaded with a couple of these biotins and polymers, you approach it to the surface, and some of these bonds will form. So you will form a complex, and so you will have a few molecules right kind of tethered in between the cantilever tip and the surface. And then you start slowly retracting, and one after the other will rupture, and eventually you will be in a situation like that only one is left, just one molecule. I was incredibly fascinated by this possibility to measure then the force, just via the deflection of the cantilever here, the force which you need to rupture one molecule, right? To drag the ligand out of the binding pocket. And so our IFM friend, yeah, please.
Excuse me, how did you do this simulation?
Not yet a simulation. That's reality so far.
Okay.
But I will get to that. There will be a simulation on the next slide. Actually, this, and I will explain in a second, right? There's no magic behind that. It's simple. But so far, I was talking about reality, not simulation. And this is what they can do. And they can measure the force which they need to drag out the biotin out of the binding pocket of the protein, the streptavidin. They don't see what's going on, of course, but they can just measure this force. And that's where they approached us and said, why can't you simulate that? Now we go to simulation. And we said, yeah, wonderful, because here we can control how fast we want to go, right? If our computers are too slow, we just pull faster, right? How do we pull? Answering your question. So in the simulation, we keep the center of mass of the protein fixed. Yeah, we don't want to drag the protein with us. And then we subject that atom here, this oxygen atom, which is precisely the one which is here connected to the, chemically connected to the polypeptide, to the polysaccharide, to the polymer. So we subject this atom to a harmonic potential, right? Just one additional term in the force field, if you want, which the minimum of this harmonic potential is just located where the atom currently is, and then we move the potential as slowly or as fast as we want here upwards. This is indicated by this spring. So we don't have a spring in our simulation. We have just a harmonic potential, which has the same Hookean law, and we move this potential upwards, either slow or fast. I'll get back to that. Yes, please.
Why do you insist on just actually pushing the single oxygen atom instead of the center of the mass of the molecule?
Very good question. Because our experimental friends cannot pull at the center of mass. There is nothing to be fixed at, right? So what they do is they attach chemically this polymer to exactly that atom. And what we want to do is to mimic the experiment as closely as we can. Actually, at some point we found out that this polymer interacts with this loop here. So in a new round of simulations, we even included the polymer to also have these interactions, and only then connected a Hookean harmonic potential. Right, good question, thank you. You might, by the way, ask the same question, why then do we fix the center of mass of the molecule? I think this is good enough approximation, but strictly, and again, in a new round of simulation, we had the whole tetramer and did exactly what's shown here, right? Not much of a difference, but just to be on the safe side. We always strive to be as close as possible to experiment, right? Thanks for the question. All right, so what comes out? Well, nice simulations, right? So here we can see this nice spring again symbolizing our harmonic potential, and we move it slowly to the right. And the curve here in green shows the actual instantaneously applied force, right? Which of course changes because the position of this spring here changes. But you can see a complex landscape of forces over time. And the largest one of that would be the one which is measured in the experiment. In the experiment, they don't have this time resolution, so they only measure the largest ones. And this is to be compared with the experiment, right? And you can probably just on the side also notice, if I run that again, that this thing doesn't just stay in the binding pocket, overcome a barrier, and then be out of the binding pocket, as many textbooks may suggest, yeah, and ligand binding, yeah, bound state, unbound state, that's it. Michaelis Menten comes from there. No, it's way more complicated, right? There's many intermediate states. At some point, you probably will see that this molecule kind of still maintains some interaction with the loop, although it has already left the binding pocket, like here now. Yeah, so there's dozens of different configurations of the whole molecule traversing kind of out of the very complex binding pocket. And one H bond, that's the main interactions which go on here, H and water bridges, rupturing after the other, right? Very complex process, which in your textbooks end up just one energy minimum barrier outside. Yeah, I hope you can appreciate what oversimplification that actually is. Yeah.
Sorry, actually we consider the binding, for example, in this case, the ligand binding as a reversible phenomenon.
Yes, correct.
But in this case, that for example, it's a PMF calculation, it would be fully reversible computational process. So
did you say reversible or irreversible?
Irreversible.
Irreversible.
Yeah.
I wouldn't call it irreversible. What I would call it is non-equilibrium. So it's a if we pull very fast, it's non-equilibrium.
But actually, we are considering the structure of the protein somehow the rigid one. It's not.
No. It's fully.
It's not completely freeze, but we are pushing some pressure or some external force on the center of mass of the protein.
Yeah.
To be completely fixed. So it means that we are controlling the conformational change of protein somehow. So how we can actually
Yeah.
directly relate this kind of bind? It's a good approximation
Yes.
for sure, but it's not completely the same as the realistic ligand binding in the real world.
Okay, yeah. So two issues. One, you're right, it's not completely realistic. This is why I mentioned before, in subsequent simulation, we simulated the whole tetramer and also ligands in there and then fixed these parts here. That is then way more realistic. We are not fixing the center of mass anymore, but we just fix those polymers which actually in reality are also fixed to a surface. It turned out, along our expectations, that doesn't make much of a difference, right? So fixing the center of mass of the protein was okay. Fixing the center of mass of this thing here, of the ligand, makes a huge difference. It is not okay. So there we have to be really careful to be as close as experiments. So that's one part of your comment. The other, again, regarding irreversibility. Here I would not agree. I would say, if we are not super slow, then we are far from equilibrium. That is true. We are in a non-equilibrium process, but we are reversible by means, if I would cut this or switch off the harmonic potential, our ligand would bind back. And that is, to my understanding, the meaning of being reversible, right? So in this case, I would say it's reversible, but not equilibrium. Agree?
Yeah, I agree.
Great. Now what comes out if I compare the two experiments, and this is shown in this graph, which I really love. So here is a summary of many of those simulations and many of experiments. And many as a function of how fast we pull. This is called the loading rate. Yeah, remember, we can retract our harmonic potential as slowly or as fast as we want. And going to the right is always faster, more and more non-equilibrium, whereas here on the left we are approaching equilibrium or quasi-static processes more and more. Blue is the experiment, and green is the simulation. And you can see, I think it nicely agrees, right? So, and I would claim there is no fit parameter in there. Not a single one. Either it fits or not. Now, the expert may say, wait a minute. In our force fields, we have tons of fit parameter. Yeah, true. But they are fit either to the solution of a Schrödinger equation, ideally, or to other experiments. So I'm not aware of any force field where only a single atomic force microscopy result has entered, right? In that sense, they are parameterized once and for all. We take this force field off the shelf, ideally several of them, try and compare. And then either it fits or not. In that sense, I think it's fair to say that it's really a parameter-free agreement. Yes?
So do you remember what force did you use for the simulations, amber or charm?
No. No. I think we use two of them. And the most which we are most frequently using is charm 36M and amber, the newest amber version. But I think I would have to look it up, or you want to look it up. One of them I think we used. And again, ideally we always use 2 or 3 different force fields to see if all is fine. Okay, so we're already quite advanced in time, so I need to decide a little bit what I want to share, which of the 4 stories I was promising, and which probably I cannot keep, which we want to share. I think maybe as a biological application, I would like to expand a little bit on that. Maybe I go a little faster than originally planned, and maybe I skip half of that. Let's see how far we go, right? But who wants to see a little bit of antibiotics biology? Is that something of interest to you? Oh, not so much. Huh. 1, 2. Okay, let's make the story very short, and then who wants to see shock freezing ribosomes? That's a few more. Are you mostly interested in solvent shells then? Ah, okay, yeah. Constant pH, I don't have many slides. Oh shit, yeah, that's that was a miscalculation. Ah, okay. Let's see how I can make things go. So let's go very quickly through antibiotics maybe, and then that is something which of course has to do with ribosomes, right? Well, not of course, but this type of antibiotics which we are looking at blocks actually, or seems to block, I should say, the exit site. So how do ribosomes work? Crash course. That's the ribosome. That's the kind of blueprint, the messenger RNA, which decodes the sequence or encodes the sequence of amino acids. This is the growing protein, a nescent chain of the protein, which is pushed through a tunnel towards the outside where it folds. And this is one of several transfer RNAs, tRNAs, and they carry one amino acid on this side, and this amino acid is exactly the one which, according to the genetic code, fits to the triplet of nucleic acids here on the other side, which then bind to the cognate triplet of the RNA. So this is actually how the genetic code is implemented mechanically. A crazy thing, right? Wonderful thing. How could ever this have evolved? Rest assured it did. This comes in, delivers the new amino acid, if it stays and fits here. A peptide bond is formed, and then it moves on to the P side and to the E side and eventually leaves, again, the ribosome, and the next tRNA comes in. That's how it works. So what colleagues of us did is they solved the structure of the whole ribosome again, but now with an antibiotics bound, which is called erythromycin, and this is bound right in the exit tunnel. Sorry, that's a different orientation. Now the exit tunnel goes down. This is the A side tRNA, which is just about to deliver the amino acid here. So this is, the red is the antibiotics, and the green is actually part of the growing peptide going through the tunnel. And if you pay very close attention, you would see some green electron density already going past the antibiotics. So if you thought, like many of us thought originally, that this antibiotic is just a cork in a bottle blocking the exit tunnel, then you are wrong. No, it's somehow different. So how is it? And this is where I need to cut a long story short now, according to this vote. What happens is, if the antibiotics here in red binds to the exit tunnel, it deforms the tunnel walls. Ribosomal RNA changes the structure. It's a conformational change. And this conformational change propagates upwards towards the active site. This is the active site, which is called the peptidyl transfer center, PTC. And it kind of pushes it back to the left a little bit compared to the unperturbed structure. And because the phosphate backbones of the RNA are negatively charged, as soon as a positively charged amino acid comes along, this is either lysine or arginine, this is also pushed away a little bit from where it needs to go. It needs to be attached to the growing chain here. That's a symbolic representation of all the amino acids growing here. It needs to carry out a nucleophilic attack to form a peptide bond to get attached to the growing chain. And if you enlarge the distance between the two, and this is what happens here because this guy is drawn to the left, pulled to the left, this bond cannot form anymore, right? So this is what happens as a result of this antibiotic. And since then, these are just details which I always skip. But since then, we have looked at many different antibiotics mechanisms in the ribosome, and all of them share this property that somehow there's an allosteric interaction which propagates to the active site and perturbs the stereochemistry of the active site. And that's what ultimately stops or slows down dramatically the work of the ribosome, the synthesis of the nascent chain. So this is how some of the antibiotics actually work, and we didn't know that before. And it always hits the active center, even if the actual antibiotic binds, I should not say kilometers away, but quite a distance away. Fun fact for those of you who plan to take antibiotics or did take antibiotics at some time, this is a commercially available antibiotic, so it's in the pharmacies. And you may realize it's important that it binds to the ribosomes of the bacteria and not to your ribosomes, right? That's the whole idea of antibiotics. Otherwise, it's called a poison. And it binds actually much stronger to bacterial ribosome exit tunnels, but only due to one additional H bond, hydrogen bond, which is formed in the bacteria and not in your ribosomes. So the difference between life and death, both for you and the bacteria, is just one single hydrogen bond. Yeah, think about it when you take next time antibiotics. Okay, I was promising to rush through that. I hope I just was able to give you the kind of the gist of of what we are going. Yeah, impressed. Yeah. Okay. This is what we can do, was an example to study biological processes and get a better understanding of what's going on. My next example does not directly study biological processes, but experiments which are set up to probe biological processes. Kind of our AFM was already kind of that, but I would like to show you one because I find it very nice, and we can also rush a bit through it, which relates to cryo-electron microscopy. So in cryo-electron microscopy, most of the structure which come up now, or increasingly more, come up rather not from X-ray scattering, crystallography, but from cryo-EM. And what they do is they shock freeze their samples, in this case also ribosomes, and take pictures when the ribosomes are then in vitrified ice, right? And these are these ribosomes, yeah? These are actually how they look at, and from that they get high resolution structure. Unbelievable, but it works. And one thing what they very often do is to classify these single images into different structures. So you can imagine that the protein does its work at room temperature. It has all the conformational motion. The ribosome does, like the two halves of the ribosome go this like all the time, and many other conformational motions. You remember the movie I showed you? Yeah, that was actually a sequence of different cryo-EM structures, of different, yeah, I was tempted to say instances in time, which is not exactly true, but different snapshots of the structures at room temperature, which are just shock frozen in this particular conformation, and then you can classify them, right, into different conformers. That's how it works. And the question actually is, in this process, they try to make it as fast as possible. So what they do is they take a grid, and each of these holes in the grid is, I don't know, a few micrometers in size. I don't even know how large it actually is, maybe a little larger. And this is one of these holes from the side. If they first put it or put solution with ribosomes on it, then they get this kind of thin film of water spanning the hole, and immersed in this water is our sample, the ribosomes, right? That's the objects of interest. And then they take the whole grid and flick it as fast as they can in milliseconds into liquid ethane at 90 Kelvin or so to really shock freeze this thing here. And when I asked my friends how fast is that, they told me, yeah, it's about milliseconds. But then if you think about it, the milliseconds is how long it takes for the whole grid to be submerged. If you put yourself into the shoes of one of those ribosomes, it may be much faster, because at some point the liquid ethane will be in contact with the film here right in the vicinity of our ribosome, and then it will probably extract the heat quite quickly. And we looked at actually how fast is that, and it turns out it's microseconds, sometimes even faster. And there's one question in the field. The question is that fast enough to really capture the protein conformational, room temperature conformational ensemble unperturbed, right? If you remember, or if you recall the free energy landscape, for example, propagated by Hans Frauenfelder, which governs the motion of proteins, a super high-dimensional free energy landscape, and proteins hop from one free energy minimum to the other over these barriers, zillions of minima. What that would mean is that you cool fast enough such that if you are in this well here as a protein, you remain there at cool temperatures. Yeah? You have no chance to go over barriers. That would be instantaneous shock freezing. But of course, even a microsecond is not instantaneous. In our hands, it's actually pretty long. It means a week of computer time. But during that time, of course, it may be that some of the proteins overcome some of the barriers and kind of focus at the lowest minima, right? That might happen. So the whole ensemble will be kind of narrowed down because it moves towards the lower energy minima due to the Boltzmann factor, which forces them to go there. And that was exactly the question we were raising, right? How much of that does happen within a microsecond? And thereby, how reliably does our shock-frozen ensemble, which we get from the cryo-EM, actually reflect the motion and the structural diversity of proteins at room or body temperature or ribosomes. And so we carried out these shock freezing simulations. You can do that. So we mimicked, again, as closely as we can, the cryo-EM simulations. And I just like this movie because it really shows how room temperature motion, here's the temperature, how this motion actually gets frozen in and into a certain structure. And we did it not only once, but many times, 41 times. I always told my student, do it 42 times, please, but he didn't know this book. And so 41. And we did that at different, some of you know that. And we did that at different cooling rates, right? And then just made a statistics, and I need to skip briefly over that, how much of this ensemble has been, or how much each ensemble has been narrowed down during cooling. And all I want you to say is that, to see is that actually, yes, that's on the y-axis is always for these different attempts, the width of the ensemble, and on the x-axis in each window is the cooling temperature, how it goes down, and all that goes down all the time. So we do see a certain narrowing, but not to zero, of the ensemble. And the bottom line of that is good news for our cryo-EM friends and for us. It's about 20 to 30% of the conformational heterogeneity at room temperature is actually lost. So that's good news for our cryo-EM friend because it's not so much. They capture the majority of the room temperature fluctuations. Good for them. But good for us, it's not 100%, so we can help, right? So we can say, all right, take your protein of interest, we simulate the shock freezing experiments, and we tell you which motions get frozen in, where does the ensemble getting narrowed down, and how you can calculate back to room temperature to get a better estimate of this ensemble from the cryo-EM. So there we can help. Okay, so that would be something we can only learn when we combine molecular dynamic simulations with experiment. Now, more of you were interested in how do we calculate thermodynamic properties of solvation shells. So looking at the clock, yeah, I probably, if that's okay, I spend 10 minutes on that one, and then I have a short glimpse of constant pH. Sorry, no more. I didn't prepare more slides on that one. But it's all published, so you can look at it. And okay, I'll get to that. Spatially resolved solvent shell entropies. First of all, why the heck should you be interested in those? Well, if you think of protein folding, you may know that actually if you fold a protein, its enthalpy contribution to the free energy, you all know this equation, I think, right? The free energy, which ultimately governs equilibria, has an enthalpic part and an entropic part here. And the enthalpic part is very favorable towards the folded state because you have all these intramolecular interactions, H bond, van der Waals contact, you name it, electrostatics, of course. So folded protein is way more stable, hundreds of kilojoules per mole compared to an unfolded protein. Yet there is the entropic part. And the entropic part is working against that because a folded protein has a very low entropy, and there's a minus sign. And that means low entropy means high free energy contribution of the entropic part, and that means unfavorable. So the protein would love to stay in the disordered unfolded state rather than in a highly ordered folded state. And the enthalpy is there to overcome that. It turns out that these two are almost the same except for a few kilojoule or kcal per moles, which then, for the folded proteins, make it actually fold. But it's a really tug of war of two large quantities, and it's just that enthalpy is a little larger for folded proteins. What does that mean? Well, the T is the temperature here, and obviously, if you would raise the temperature, you would increase the weight of the entropic contribution, and then entropy should win, right? That would be our prediction. And that's indeed true. You all know that if you heat up a protein, if you cook an egg, for example, it unfolds. Stiff if you could cook it. It unfolds and then it intermingles and all that. Okay, so that's called heat denaturation, right? So also if you get fever, that's one of the unfavorable things which happens. Your proteins unfold, not good for you. So that works, our theory works, seems to be something against it. Now it makes another prediction. If you reduce the temperature below room temperature or something, then obviously enthalpy should win even more, and that should stabilize the protein even further. So the colder you are, the more stable your proteins should be against unfolding. And unfortunately, this is just not true. You can have as nice a theory as you want. Experimentalists tell you, sorry, wrong. There is something like cold denaturation. Most proteins, not only exceptions, but most of them actually also unfold if you cool it down. What the heck's going on there? We missed something. And what we miss is actually the contribution of the solvent. We missed that the hydrophobic effect plays a crucial role, and that's the action of the solvent, plays a crucial role on the folding of a protein. And it just happens that the hydrophobic effect, stabilizing also the folded state, gets weaker and weaker the colder the water is. So I'm just saying that or mentioning that to motivate you. We can't forget the solvent, and the thermodynamics of the solvent, specifically its entropy, is crucial not only for protein folding, but all the motions, conformational motions protein undergo. And in our field, this term is mostly forgotten. Of course, it is included in the simulations. Don't get me wrong. That's one of the reasons why I really love explicit water simulations. It's included there, correctly mostly. But we don't get numbers, right? We don't know how much the entropy of the water around a particular amino acid actually contributes to folding. And this is what this project actually was aiming at. This is what we wanted to find out. We wanted to have a kind of a map of water molecules around a protein. This is Grubmüller, just a small example, where we could assign here color-coded a particular thermodynamic quantity like enthalpy or entropy to each single water molecule to understand how that contributes to the stability of a protein. And I should probably mention that, of course, we are not the first to do that, but I should say most of the methods available, if you think of thermodynamic integration or free energy methods for the expert, they just assign a total entropy to the whole system rather than focusing on single water molecules. So you don't have spatial resolution. And it's also not so easy to define that. I need to skip that. But we managed to do that. And I want to give you just a gist of what we are doing here. So if you know quasi-harmonic analysis or Schlitter's formula, some of you might know that, to calculate the entropy of a protein, what you essentially do is you take a protein, you simulate it, it fluctuates, and then fluctuating means that you have a certain conformational density distribution in this high-dimensional n dimensions, x, y, z, 3n times. And this is here symbolized here like that. And because it's a folded protein and doesn't depart much from its average structure, you have a very localized conformational space density describing that protein. That's this ellipse here. And using principal component analysis, sorry if you don't know that, but that is a way to describe the shape of the ellipse and essentially also to describe the whole density here in terms of a multivariate Gaussian. All I want to say is that works wonderfully for solvated proteins because they have this nice conformational density shape and can be car plus times. In the solvent, it looks totally different. All the water molecules can zoom over in the whole box, and so you just can't play the same trick on the solvent, and that means you can't calculate the entropy of the solvent in a straightforward way. And what we came up with is the good old Gibbs factor just the other way around, right? What do I mean by that? What we came up with is the following idea. Let's say we have just two molecules, two water molecules, and they are move just in one dimension, such I can draw the total conformational space. That would be this one here, right? Only two x coordinates. And let's say they move happily back and forth. That would be the green trajectory here, moving in this space. Now, observe that when we cross this line here, from here to here, then these two molecules have switched location, apparently. And the situation is just like before, except that now number one is left and number two is right. But the physics, well, water molecules don't actually carry numbers, right? It's just an artifact of our simulation. So the physics is totally unchanged. In the configurational space, that translates into that the whole density here on this upper triangle is exactly the same as the density in the lower triangle. So rather than letting the molecule move over there, we now just renumber it, and by that we mirror it back to where it should go, to this lower triangle. And that is indicated here, right? We just swap the numbers. Two important lessons here. Number one, that reduces conformational space. You may say factor of 2 doesn't impress me. Yeah, but if you have 100 molecules, this factor is 100 factorial, and that should impress you. So the sampling space is suddenly 100 factorial smaller than before. That makes it practical. That makes it workable. That's number one. And number two, what we do now is we essentially, we localize our water molecules, right? So if we color of the water molecules and use an original simulation without this trick, then of course every color goes everywhere. But if we do this relabeling, as soon as a water molecule wants to go into the region of its neighbor, the numbers are swapped and it's put back, so to say. Except to say that not the molecule is put back, it's just the labeling. And I'm insisting in that because that tells you it doesn't change the physics. It's just the labels. And so the physics is totally unchanged. And that makes a kind of a small piece of conformational space, which then we can use to calculate the entropy. And if you really want to go into that, we need mutual information expansion. No time to explain that. But the result is it works, and we can now calculate entropies of water molecules. And moving quickly forward, to just show you one example, which we did recently together with Jussi Taipale. This is a transcription factor protein on the left, on the right side here, which binds to a specific sequence of DNA and regulates transcription that way. And the funny thing here is, of this particular binding factor, it binds to two different sequences only, and only to these two, with about the same free energy or affinity, this one and this one, AC and TT. And what they know from calorimetry and from X-ray structure analysis is that the binding here is mostly enthalpic, governed by very favorable enthalpy, and this one mostly entropic. That's very unique, but it's a particular property of that system here. And they were wondering, why is that, and where is this entropy? And the idea was maybe it's in the water, not so much in the protein, because the protein structures in both cases look perfectly alike. And so we looked into the water with that method and found, now color-coded, much higher values here in blue than here, lower values, sorry, in blue than in red. And so here indicating, yeah, the combined entropy of all those water molecules at the interface is actually significantly higher, and so we were able to quantify that one. All right, so much for solvent shell, or in this case, interface, water interface calculations of entropy. You couldn't do that with standard GROMACS before. Now you can do that. If you want to do that, have a look at our homepage and we can provide you with the code. All right, and then I was promising to do some constant pH simulations. And as I said, we only have very few constant pH simulations, but that is something which I think in recent times became very helpful. It's been implemented in GROMACS now, actually in two different flavors. One is by Gerrit Grönhoff, which is called charge scaling and works, and the other is by us, which we call Hamiltonian lambda dynamics. It also works. So what you can see here is, for example, carboxy groups and the protons with it. And I have drawn in this movie the protons according to their probability to be there, right? You can see that sometimes it grows, sometimes it shrinks. That's exactly what happens in such a simulation. I should say, the trick what we are doing is we extend the Hamiltonian, or the force field in this case, of the protein by this terms here. And those of you who do free energy calculations know that that's essentially free energy perturbation, where you morph one system described by this force field into another one. And we do the same in these constant pH simulations, except that this lambda here, which, as you can see, shifts, if you go it from 0 to 1, shifts from one Hamiltonian to the other one. That one is not now shifted by hand like in a free energy calculation, but is a free parameter or a free dynamic variable. So it moves along the same laws, Newton's laws actually, as all the atoms also move in the system. And for every titrable group, like in this large system where you have tons of titrable groups, we have one of these lambdas, and thereby each lambda always moves between 0 and 1 and let the proton appear and disappear. And the nice thing about it, this mechanism, if you do it in a strictly Hamiltonian way, like here, like we are doing it, it guarantees you that it follows the thermodynamics of the real protons, right? That means if the pKa is very high, you have very low probability to have the proton, except you go to a very low pH, a high pH, and so on. So you get the correct titration behavior. And for the experts, if you don't do constant pH, but conventional force field simulations, you have always to worry about the protonation, particularly of the histidines, right? So, because they have pKa about 6.5, so depending on the environment, chemical environment, they are either protonated or not, or even worse, they may change all the time, right? And constant charge simulations don't allow you to do that, or only with quite a bit of effort. So if you switch to constant pH, that does that for you, right? If you protonate it wrongly, it will correct it during the simulation automatically. There's a few more remarks I wanted to make. I don't have the time. Just to mention, it's fully implemented also in GPUs, so it's fast, and it uses a different electrostatic solver for the experts now, not PME, but fast multiple methods, which has a few scaling advantages. You can probably go into that if you want. What you also can do with constant pH simulations, and then I'm soon at the end of my talk, you can do titration. You set externally the pH you want to have, very much like you set the temperature and the pressure in a conventional simulation. In addition, now you can also set the pH. And then you just count how often your proton, or your titrable group is protonated, and that gives you the fraction of protonation and eventually this titration curves. Can derive the pKa of this individual titrable group, and you can do that for any group you want in the protein. We did that for a number of test systems, and it turns out, compared to experiment, NMR is able to measure that. You get it about half a pKa unit correct, right? That's about the error, the uncertainty we still have. All right, so that's for constant pH. We deliberately, we put a lot of effort, and if you want to know more about it, ask Carsten Kutzner, who will be around shortly. We deliberately did it so that mostly you just have to turn a switch, and your conventional GROMACS simulation turns into a constant pH simulation. It's about a factor 2 to 3 slower. That's the price to pay. We hope to work on that. But other than that, it should actually work pretty well. All right, that brings me to the end. I see I'm now moving into the coffee break. I should not do that. But I just want to leave you with one more almost philosophical thought. I have seen there is no philosophist in there, but still I think it's a fascinating thought. What we have done, not we, but our community, over the past like 70 years or so, we have started with the Schrödinger equation and started applying then the equation to molecules, like a water molecule in the 50s or so. And later on, Martin Karplus and others started to simulate proteins, first in vacuum, which is of course nonsense, but still a good start, and then in water, except if you want to do mass spec, then it's important. You have seen a number of those systems. I skipped a whole virus, which we did a while ago, or a whole membrane budding. That would be an organelle of a neuron in this case. Microtubules, whole microtubules we did recently. And we get to larger and larger systems, we get to longer and longer timescales. But within the limits of our approximation, or 3 levels of approximation, which I have shared with you, what we are essentially doing is solving the time-dependent Schrödinger equation. And I have no idea where that goes into bacteria or frogs or whatever. That will probably take a while. But nevertheless, I find it absolutely fascinating that we really walk all the way upwards with just the Schrödinger equation. We just solve this super simple equation, and that's what we get, including ourselves. And I think this is a fascinating talk. I realized just at a congress recently that 90%, I did a statistics, of biologists don't agree with me. They say, wow, no, this is more to us, right? Consciousness and what know I, right? And I would, just to challenge you a little bit, I would challenge that and say, no, I don't believe that. I believe the Schrödinger equation is enough if you go all the way upwards. It's a lot of complexity, it's a lot of emergent phenomena, but that's all you need. And with that, I think we live again in very fascinating times on this journey. Let's see how far we get in the next 10 or 20 years, and looking forward to your questions. Thanks so much. If there's time for questions. Sorry for filling up the time so much. The chairman needs to decide.
If you have questions?
I'm curious to know about the constant empty simulation and its difference between the, let's say, the ancient method for determining the hydrogenation state or protonation state of the amino acid. For example, previously we could do something like this with the what if or the Pro-pKa to just determine the protonation state of the amino acid and do the simulation.
Yes.
What would be the difference to pay the time cost effective for this kind of empty simulation?
Yeah, excellent question. Thanks for that one. First of all, I don't want to put much blame on Gerd Vriend, but what if can be wrong? What if it's wrong, right? So it's an empirical estimate. It looks at nearby charges, it looks at H bonds, and empirically, by comparing with high-resolution X-ray structures in the PDB, it guesses what the protonation might be. Here it's calculated, it's not guessed. So that's number one. Number two, what if things change? What if the proton or the pKa is just about 6.5 or 7 and you simulate its pH 7, then the proton goes on and off. What do you do then with classically or established simulation? Do you put half a proton on it? I don't know. Constant pH does that for you. Also, if you have a conformational change, then very easily the pKa of certain groups may change. If something moves into a membrane, an arginine moves into a membrane, it may even get protonated or something, right? And what if will not tell you that because you're right in the middle of a simulation? Constant pH will take care of that. Yeah. Also, I haven't expanded on that, but what you can now look at is the coupling between titrable sites. Yeah, one gets protonated as a result that couples to another one, either directly via electrostatics or indirectly via conformational motions. Also, that you can study now. So it's kind of you don't have to worry anymore, except for computer time.
Yes.
Well, we hope to get faster. There's one final trick which we have up the sleeves, which should get us very close to normal GROMACS. So then you don't have to worry about time anymore. Mm-hmm. We have, by the way, published that, so I hope I have the two publications somewhere. There. So if you want to, I guess you got the slides anyway, right? So these two publications of this January, they describe this implementation and how to carry out these simulations. Or you look at our homepage. Yes.
In one of the slides, I saw that you compared the experimental MD simulation and also the Brownian dynamics results.
Of what?
The Brownian dynamics in the graph that you showed for the comparing the MD results and the experiment.
You're referring to constant pH or?
No. It was about, as I remember, antibiotics discussion. I don't remember exactly.
So the erythromycin binding to the ribosome?
Maybe yes.
No, it was the
I
spring
think it was the
one.
water.
Yes.
The what?
The spring thing, where you have the beads and you have the...
Oh, the AFM unbinding.
Yeah.
Smokers. Yeah.
First, I'm curious to know about the Brownian dynamics and the GROMACS.
Yeah.
Is it completely compatible to do with the GROMACS or no?
The Brownian dynamics, you mean you don't have explicit water or what?
No, it would be the implicit water for sure, but kind of, as I recently studied, it seems that integrator of the BD as a Brownian dynamics has been implemented in GROMACS.
The Brownian dynamics of
Yeah.
GROMACS. Okay, I get it. I'm not a fan of it. I would guess the results, it does have implicit water, right? So most of the cases. And so all the effects of the water molecules, I should say many of the effects of the water molecules on the protein would not be included. And you would probably get different results. But I haven't tried it out, I don't know. In my simulations, I just made the rule, we want to have the water in there, right? So it takes us maybe another factor, 2, 3, 4, 5 more computer time, but then we know that we have the water in there. And Brownian dynamics, depending on the force field which you have, right? So there's a whole zoo of different force fields for implicit water models. You may be more or less accurate, but I just don't want to worry about these things. So I haven't tried it out, so short answer, I don't know what would come out. Okay, let's leave it at that. Thanks very much.